Hello, everyone, and welcome to this first session of 2026 of the Athletic Action Journal Club. Um, as you know, this is open to all researchers and scientists, but we especially like to use this space to give voice to the young researchers. So for those connected, please feel free to ask your questions in the end of the session and like to interact as much as you want. And for the senior members connected, please remember about this opportunity for your young lab members and invite them over for the sessions to come in the next months. Christina will introduce our speaker. Over to you, Chris.
Good morning, everyone. We're super happy to have with us Dr. Debanjan Mukherjee, who's currently a postdoctoral researcher at the University of Freiburg in Germany, studying epigenetic regulation of heart failure. He earned his PhD from the Institute of Cardiovascular Regeneration in Frankfurt, focusing on transcriptional regulation of aortic aneurysms. And today we are super happy to have him and also Nuno Guamairas Gamboa in presenting their recently published article in Cardiovascular Research entitled Transcriptional Regulation by TBX18 in Smooth Muscle Cells is Essential for Normal Aortic Development and Homeostasis. Thank you so much for being with us, both Debanjan and Nuno, and we look forward to your presentation.
Thank you, Christina, for having us in here. I hope you can hear me and see the slides. Okay,
Yes.
All good.
Go ahead.
perfect. Okay, so good afternoon, everyone. To begin with, I think everyone is mostly aware of the cardiovascular system, but just to remind you about the whole physiology of the mammalian aorta, as we know, during systole and diastole, there are fluxes of blood ejecting out of the ventricle. But at the level of the capillaries, when you see that the flow of the blood is almost uniform, and the reason behind this so because of the aorta, which expands and contracts accordingly to regulate the pressure of the ventricles and to dissipate it to the capillaries. And this is possible by the expansion of the contraction during the systole and diastole, and thanks to the specialized architecture of the aortic wall, which is comprised of the media, mostly the smooth muscle cells, and the other layers like tunica intima or adventitia. So mostly these smooth muscle cells in the tunica media helps in the expansion and contraction, allowing the aorta to dissipate the pressure uniformly throughout. And any certain kinds of be it physical or genetic or any mechanical kind of problem or any kind of defect in the smooth muscle layer of the tunica media can lead to serious disease, for example, or disorders like aneurysms. So what are aneurysms? Vascular aneurysms are pathological dilation of the blood vessels that leads to vascular rupture, and aorta being the biggest blood vessel, aortic aneurysms are the most frequent and the deadliest kind of aneurysms, accounted for 1 to 2% of the deaths in developed countries. And as you can see, the aortic root bulge is a typical manifestation of the aneurysms. However, depending on the position, it can either be thoracic or abdominal. From a clinical viewpoint, when we look at aneurysms, and aortic aneurysms can be either idiopathic aortic aneurysms, which do not have any evident genetic component and often acquired by some risk factors. For example, smoking is considered one of the risk factors. But on the other hand, when we look at the familial or heritable aortic aneurysms, they have a clear genetic component, and these are heritable, and based on these clinical heritable aortic aneurysm, it can either be syndromic or non-syndromic. As the name suggests, syndromic aortic aneurysms have various manifestations, and also many of the important genes have been uh, uh, regulated in these syndromic aortic aneurysms are mostly related to the extracellular matrix components. On the other hand, the familial but non-syndromic aortic aneurysms do not manifest any pathologies as such, but important mutations that have been identified in this case have been implicated in the smooth muscle machinery. For example, mutations in ACTA2, critical genes like myosin heavy chain 11, or even transgallin. As of now, only say around 21% of the cases where the aneurysms have been linked to the genetic source, while the other cases have not been correlated with any clear genetic component known so far. And another important thing is that in these cases there is an incomplete penetrance into the familial tree, which makes it even difficult to link it to any particular genes. From here, it is so far clear that the genetic contributors of the aneurysms can either be the extracellular matrix molecules like fibrillin 1 causing the Marfan syndrome, or fibrillin 4 causing the cutis laxa, and so on and so forth. Otherwise, there is also TGF beta signaling, where the receptors of the TGF beta signaling causing aneurysms are having manifestation as seen in Loeys-Dietz syndrome, and also the smooth muscle contractile machinery upon mutations like the important genes like MYH11, ACTA2, and so on and so forth. They are causing aortic aneurysms. But what is not much known is what are the upstream regulators or the transcription factors regulating this disease, and therefore we want to study what can in the upstream regulate these signaling pathways. When we look at overall the smooth muscle contractile machinery, what is also very important to know is that the smooth muscles, and upon disease or during developmental states, uh, undergoes a phenotype switching from where a contractile, which is a differentiated state, often switches in a disease condition to a synthetic or a de-differentiated state. And also in this kind of switching, what are the transcription factors or upstream regulators regulating these are not much known, which brings us to the protagonist of the story, that is TBOX18. What we have seen is that TBOX18 is robustly expressed in the aortic smooth muscle cells from early time points till late adulthood, and therefore we hypothesize that it might have important role in aortic homeostasis and biology. So, moreover, in case of disease context, with our collaborator from Lars Magnificel's group, what we have seen is in a cohort of patients with aortic aneurysms, this particular transcription factor, TBOX18, is significantly reduced only in the medial layer from patients compared to the control aorta. In the aorta having aneurysms, there is a significant decrease of the expression of the TBOX18, and it even increases the expression decreases further compared to the controls when the aorta having thrombus, which is a more progressive state of the aneurysm. And this is only restricted, as you can see, in the medial layer, where the adventitial layer do not show such a regulation, which also emphasizes on the role of TBOX18 in the disease context in the smooth muscle cells. So what is this TBOX18, we wondered, and this TBOX18 is a transcription factor belonging to the TBOX family of transcription factors, where other family members, for example TBOX5 or TBOX20, have been extensively studied in cardiac development and in disease context, with multiple NGS datasets available and several several molecular tools which are available, making it possible to study it further, which is lacking in case of TBOX 18. So what is known about TBOX 18, thanks to Andreas Gispert's lab and many other labs working on TBOX 18, that we know that the global knockouts of TBOX 18 are perinatally lethal, therefore which precludes much of the studies to know its roles in adulthood. And so far there are only a few RNA-seq data sets available, and that too with no ChIP-seq data available. Therefore there is a very little knowledge about its direct targets. Therefore, knowing about these important information, what we wanted to see in the like to know its role, what are the major aims of the project, was to look at whether TBOX 18 is essential or required for normal aortic development, and also to assess if TBOX 18 is required even after development, say for aortic homeostasis in adulthood, and finally to identify the gene expression networks directly regulated by TBOX 18. So to begin with the first aim, so for doing so, we wanted to constitutively using a constitutively mural specific conditional knockout of TBOX 18, in this case using driven by the PDGF receptor beta CRE. Um, as you can see, after ablation, the T-box, these PDGF receptor beta Cree line specifically marks the mural cells of T-box 18 expressing mural cells without hitting the other T-box 18 expressing populations, for example, the cells of the epicardium or the cells of the ureter, showing the specificity of the Cree. And now, knowing so, when we wanted to ablate TBOX 18 in the mice, this was done in collaboration with Professor Sylvie Evans' lab in UCSD, US, and the data showed like for after knocking out TBOX 18 conditionally in the mural cells, though the progeny were born in normal Mendelian ratios, there was increase of the death of the conditional knockouts from early adult time points, and by 4 months of age, more than 50% of the conditional knockout cohorts were dead. And therefore, we wondered why might be such a death, and the animals which still at 2 months or 8 weeks time point, as you can see, were typically weak compared to the controls. The conditional knockouts were showing less amount of body weight, as well as increased systolic blood pressure, and therefore we wondered, is there any problem also with the arteriogenesis? And to do so, after doing the aortic angiography, what we have seen compared to the wild types, where you can see a nice straight aorta, in case of the conditional knockouts, we see clear aortic tortuosity, which also have been quantified in here. And this is one of the problems that often arise from defects in early arteriogenesis. Now looking at it, since these animals were super weak and most of the animals died upon older age, the ones which still managed to survive were showing even pronounced effects of aortic dilatation and aortic aneurysm like phenotype, where you can see compared to the controls, where there is the aortic root normally size. In case of the conditional knockout, there is a pronounced aortic root dilatation with fibrosis, and also the aortic wall integrity is not preserved, and it shows often breakage of the elastin lamina, and the adventitial layer is also disrupted, showing that how TBOX 18 is essential per normal aortic development from early embryonic time point. Now, after knowing so, we wanted to see whether T-box18 is required after development for aortic homeostasis in adulthood. And to know about its role in adulthood, in this case, we have used an inducible smooth muscle specific conditional knockout. This is driven by the myosin heavy chain 11 Cre ERT2, which is an inducible Cre. And secondly, what we have introduced is we have studied this model in presence or absence of a fibrillin 1 mutation, which recapitulates the Marfan disease, which predisposes the aorta to form lesions and undergoes aneurysm formation, so having a genetic component to it. So the experimental groups were either only control animals, only T-box18 single knockouts, then only Marfan, and the double mutant which has the not only T-box18 knockout but also a Marfan background, which is the double mutant, and I will be using these terminologies for the subsequent slides. So what we have done is upon tamoxifen induction, we have looked by non-invasive echocardiography at different time points as shown in here, or by histology at different time points. What I now show you in here is the echocardiography results, and what you see even before even tamoxifen induction, where there is only the control group or the Marfan group, we see an increase of the overall aortic root diameter, and upon time course echocardiography, we see a trend of increasing aortic root, which is normal because the animals are all growing. However, if you look closely, from 6 to 8 months time point, only this double mutant group are showing an increased amount of aortic root diameter, and as you can see also from these representative images that compared to the controls or the single knockouts, double knockout shows pronounced aortic root dilatation and also quantified as in here. Now, to further substantiate these in vivo findings with histology, we saw similar trend compared to the controls and the single knockouts. The double knockout shows significant increase of the aortic root diameter. What is even more interesting in here is that only during this time point from 6 to 8 months, it coincides with the wave of lethality that we see in these double knockout mice, where the double mutant mice, where the other cohort, other controls or the single mutants are not being affected, which was interesting. And therefore we wanted to look into closely what happens at the aortic integrity, as I can show you here with the help of the elastin and the TD tomato, which marks the smooth muscle layers, that in case of controls, the elastin layers are well organized with smooth muscle almost equidistant from each other, having separate concentric layers of the smooth muscle cells separated by elastin in green. But when we look at the single T-box18 knockout or the Marfan mice, we often see there is occasional thinning of the elastin lamina or occasional breaks of the elastin lamina and fusion of the smooth muscle layers on and off sometimes, but which is even more exacerbated in case of the double mutant, where there is a havoc fusion of these smooth muscle layers and also the typical thinning and loss and breakage of this elastin lamina in case of these double mutants, and also which has been quantified by either the width of the elastin lamina, the width of the individual smooth muscle layers, or even the number of smooth muscle layers, which is significantly going down. Now from here, what we can clearly see how in double mutant T-box18 conditional ablation exacerbates the Marfan phenotype. Now, to summarize so far, what we have seen is how T-box18 is expressed in the aortic smooth muscle cells, how the mural specific ablation of T-box18 from early embryonic time points lead to severe aortic root dilatation and aneurysm-like phenotype, and upon mural ablation of T-box18 from early time points leads to the lethality in the young adults. And finally, we have seen in adulthood when we ablate T-box18, it is not lethal, but becomes lethal when it's combined with a Marfan causing mutation. Now, after knowing so, we now move ahead to look at the third aim of our project, that is to identify the gene expression networks regulated by TBOX18. Now, to do so, we use a transgenic approach to look at the translating ribosome affinity purification sequencing, which is also known as trap sequencing. And the principle here is that we are using a transgenic allele where the ribosomal large subunit L10A is fused with a GFP, and this GFP only upon tamoxifen injection is expressed in the Cree of interest. In our case, it was a myosin heavy chain Cree ERT2, and upon tamoxifen induction, this ribo-GFP becomes activated only in our case smooth muscle cells, and after 4 weeks after tamoxifen treatment, we are harvesting the animals and harvesting the aorta to undergo a trap pull down, which is nothing but a pull down of the ribosomes, which are GFP labeled using anti-GFP antibodies, and these ribosomes containing the actively translating mRNAs. Therefore, after extracting the RNA and sequencing, we should be getting the actively translating mRNAs, giving us the translatome. On the other hand, as a control, we also use the total RNA, which gives us the transcriptome of the aorta. To see the proof of principle, we also look the control IP versus the input, which is the to see whether there is an enrichment of smooth muscle specific transcripts. And as you can see, compared to the control, where we see enrichment of various cell types like the endothelial cells, fibroblast, immune cells, and also some of the other cell types like the lymphatics. On the other hand, where we see in case of our trap-seq samples, there is a clear enrichment of the smooth muscle specific transcripts, showing that this technique works perfectly fine, and it's like a proof of principle. Knowing so, then we moved ahead with our actual samples. Either we have done the trap sequencing on the single knockouts, only the Marfan, or the double mutant. And what we have seen overall from the first clear view, what we can see from the number of genes up and down regulated in the translatomics, as you can see, like we had a gradient of the phenotype in case of from our morphological data. Even in the molecular data, we can see compared to the single mutants or the Marfan, when we look at the double mutant, there is overall a much increased number of transcript differentially expressed genes, and both in the up and down regulated categories. And what is striking from here is that in all the categories, we see a clear upregulated immediate early response genes, and which is also in the form in the same gradient as we see, where the double mutants has more number of these immediate early response genes, which not only more number of genes but also with increased level of their transcripts. And many of these extracellular matrix genes are also upregulated in these double mutations, also hinting towards ECM remodeling. And interestingly, if you look at the double mutant, there is a clear downregulation of this circadian clock genes and muscle contraction genes, which also hints us towards the pathological role, why we were maybe seeing such a havoc phenotype in our morphological analysis. Now, knowing so, as we saw that the immediate early genes were going up in almost all the categories, we were wondering what are these genes. And as you know, this immediate early response gene, when we look back in the literature, are some of the immediate effector cascade pathways which generally are induced upon stress and in disease phenotype, where this induction of the immediate early genes are transcription factor, which are inducible transcription factor, which carry on a cascade of transcriptional regulation, and therefore thereby affecting the effector molecules. And in the context of the aortic aneurysms, in two or more different models of totally more different gene models, for example, the fibulin 4 deficient model, EGR1, one of the immediate early response genes in our translatomics data, is shown to be regulated, and similarly how in our model we see EGR1 getting upregulated. And very recently, last year, also from a single nuclear RNA sequencing data on human aortic patient samples and mouse um, cohort, mouse patient, mouse the sample, single nuclear RNA-seq on mouse model of aortic aneurysms, also have seen this immediate early response gene going upregulated, showing or hinting towards a conserved response pathway in aortic aneurysm. Now, to see which of these immediate early response genes are direct targets of TBOX18, we wanted to look at the TBOX18 bindings in the chromatin, and unfortunately, there were no available ChIP-seq data. Therefore, we used human aortic smooth muscle cells and overexpressed TBOX18, followed by a anti-flag chip due to unavailability of direct TBOX18 antibodies, followed by chip sequencing. And as you can see, around 30% of our peaks were promoter bound, and we were super happy to see that upon great analysis, the significant terms which popped up from these genome bound peaks were related to aortic dilatation or aortic aneurysms. Furthermore, to do a motif analysis using Humor, what we have found that most of the important peaks that have been shown like for 14% of all peaks are showing the motif for the degenerate T-box 18 binding site, and also by the T-box 20, 3.5% of peaks showing the T-box 18, T-box 20 binding the T-box domain, like the degenerate mode, the motif for T-box binding, T-box family uh, transcription factor binding is shown up by 3.5% of our peaks, showing that there is a increase consistent binding of these transcription factors and the T-box 18 that we have identified from in here. Now, from here, what we wanted to see is to integrate the ChIP-seq peaks from that of the TRAP-seq from our translatomics data, and also in here when we integrate these ChIP-seq peaks with the DEGs, or in this case the differentially translated transcripts. And what we see is also that the immediate early genes are the direct targets of the TBOX 18, and in case of double mutants, even the number of genes or the number of targets also increases. Which comes, with this I will now show the validation and also some of the tracks. Like for example, the genes of the immediate early response genes like the FOS, JUNB, EGR1, and its effector protein thrombospondin is shown to be clearly bound by TBOX 18 at the gene bodies, and also validated by Western blotting. As you can see that in the double mutants compared to the controls and the single mutants, there is a significant increase of this effector protein, showing how the pathway is working. Now we wondered whether these regulation is also conserved in case of the patient cohorts, and thanks to Professor Lars Magdefissel's data cohort, with our collaborator, that we have looked at the transcriptomics data of the patients having aortic aneurysms, and what we have seen that compared to the controls, when we looked at the elective aortic aneurysms and ruptured aortic aneurysms, there is kind of even a gradient of decrease of T-box18 expression, where the progression of the disease is also uh, correlating with the decrease in the amounts of T-box 18. And on in the similar trend, when we look at the negatively regulated direct targets of T-box 18, we see upon T-box 18 decrease, there is an increase of these direct targets like immediate early response gene, for example, FOS, JUNB, EGR1, and thrombospondin, which is also in the similar trend going up compared to the controls, showing that even the mechanism is conserved in case of the patient cohort. And not only that, these are the abdominal aortic aneurysms, but when we looked at in case of the thoracic aortic aneurysm, so we were super happy to have luckily the cohort of thoracic aortic aneurysms, be it sporadic or in Marfan cohort, and in both the cases we saw by RNA scope, that is looking at the in situ RNA in situ hybridization technique using fluorescent probes and the fluorescent detection technique. And what we have seen compared to the controls, when we look at the dilated lesion areas, there is a significant decrease in the T-box 18 transcript, both in the sporadic or in the Marfan patients, also corroborating with our findings in multiple different cohorts. And this brings me to the model where we see in case of control, where you see a normal aorta where T-box 18 basically is inhibiting, T-box 18 is present and it is inhibiting all these effector molecule or the immediate early response gene, which upon T-box 18 knockout, uh, this regulation is lost, and we see increase of this thrombospondin expression of the effector protein, which is also in case of the Marfan, what we have seen that this immediate early response gene goes up, which even exacerbates further where this Marfan is present and T-box 18 is absent, where the regulation is even more exacerbated, which also leads to high amount of thrombospondin 1, which basically leads to this very strong phenotype and leading towards lethality in this in the scenario in of aortic aneurysms. And now with this, I come to the last part, which is also one of the important aspects to see how in the during this submission of the manuscript, the how the peer review enhancement makes the story even more lucid. And we were super thankful to our reviewers who gave positive feedback and suggested some experiments, for example, including the mouse experiments, where they have asked us to look into the earliest time point where, or in the embryological time point where we could detect T-box 18 expression. And in the mouse experiments, we have looked at from earlier like E10.5, where we saw still there is a presence of T-box 18 expression in different time points we have looked into. We have also been asked to give cohort clarification of the different cohorts that we have used, patient cohorts in our study. And due to some German regulation, we were not allowed to use a lot of the information due to data protection, and some of the other cohort some of the other validations that our reviewers also have hinted towards is to look at by wet lab validation in different cohorts, if or in different types of aneurysms, whether it is consistent or not. That also boosted our findings even more translationally. There were regular routine control experiments that were asked, for example, to use the IgG as the controls to show the antibody binding, and also some of the experiment with graphical rearrangements and figure rearrangement, for example, to look into different time points more specifically, with every time point specific p-value quantification, and looking into individual time points and their respective effects. So these are the overall different kinds of peer review enhancements that were asked for us to include, and I think that positively impacted the overall manuscript to make it more understandable. With this, I will bring you to the take-home messages from this entire talk, where we have found that how TBOX18 is expressed in the aortic smooth muscle cells, how ablation of TBOX18 from early time points lead to aortic malformation and lethality prior to 6 months of age, and how ablation of TBOX18 in adulthood is not lethal but becomes lethal when it's combined with a Marfan causing mutation. From a mechanistic level, TBOX18 it represses the immediate early genes and promotes muscle contraction and circadian clock. And TBOX18, on the other hand, promotes extracellular matrix production, represses cytokine signaling, and immune synthesis genes. And finally, the TBOX18 expression is reduced in multiple aortic aneurysm cohorts. With this, I'll come to the final slide of this presentation, and I would like to thank Nuno, Dr. Nuno G. Gamboa, my supervisor for the PhD, and the entire lab, including Professor Dr. Stephanie Dimmler, for allowing me to work in the Institute of Cardiovascular Regeneration, and all the members of the Gamboa group, and members of the Dimmler group, and also the collaborator, especially Professor Dr. Sylvia Evans and the group in UCSD USA, and Professor Lars McDiffisels and his team members for the precious samples and the data for to include. And finally, our Stefan Gunther, Dr. Stefan Gunther from Max Planck for the sequencing, and the group in Monzino also for the critical thoracic aortic aneurysm samples, and definitely last but not the least, the funding bodies for keeping the projects ongoing, and also for the remunerations. And thank you very much for your listening, and I'll be happy to take questions.
Congratulations, brilliant presentation, Debanjan, and it's nice to see this work explained by you. I can see we have already some questions in the chat, so I would say, Yaya, if you want to come forward and ask yourself your question, feel free to unmute yourself. Otherwise, I can ask this. Let me know. If, let's say, just in case they are not able to do this, let me ask the questions then. So, Debanjan, the first question is, as TBX18 is targeting AGR1, which regulates also inflammation and chemokine expression and probably cell migration, what about plaque development? Do you think TBX18 could have a role in this? In atherosclerosis and plaque development?
I can suggest, I can only speculate at this point that if you remember in the initial phase, I was mentioning that the smooth muscle cell exists in both the contractile phase as well as the synthetic phase. And in plaque development, we often see that the synthetic smooth muscle cells are the ones which are facilitating the plaque formation and also mobilization later on to form the CADs, and yeah, and therefore one can speculate that yeah, it might play critical roles in plaque formation as well.
Thank you for that. And a kind of related question, looking at the very nice data that you were presenting, in particular on the double knockouts, and how these, especially, like you see how the changes and this aberrant phenotype is marked around 6 and 8 months, if I'm correct. So looking at how the integrity of the aortic vessel changes in that late stage, I was wondering whether you have considered or had like any comments about like potential role in aging. Could that be something impacting? Of course, mortality raises because the double phenotype is there doing something, but like, could that be linked to aging in a way?
Um, yeah, if you compare the other controls, for example, wild type control obviously, but if you even look at the single knockouts, be it the only Marfan or only T-Box 18, we do not see much of a havoc phenotype going on. Yeah, therefore it is only even from the molecular data, if you look at the number of genes getting regulated in the each of the single knockouts is not so prominent. um, but when T-Box 18 comes along with the Marfan, then the double knockout is more showing the havoc effect and also lethality. When it comes to aging, in the single knockouts, also from the molecular aspects, we do not see any direct aging related genes coming up from the translatomics data, but there are hints, like for example there can be secondary effects through various other mechanisms, for example, the circadian clock, which can contribute towards aging, and it's as a secondary of effects more.
Brilliant. Yeah, no, I was exactly thinking about like the changes in the circadian clock, and that's why I kind of was curious. Thank you so much. I think we had a hand raised from Melody.
Yes,
If you want to
thank you
take it over.
for thank you for an exciting work and congratulations on getting this story wrapped up. I had also a question. It's more of a thought also. How come you decided to explore T-Box 18? Like, was this gene a result of a previous transcriptomic analysis in AAA, or how did you end up with investigating that specific gene? What led you to it?
Mhm. Yeah, that's a very classic and important question, and this I would really thank my supervisor, Dr. Nuno Camboa, for introducing this gene to me, and that's because in his former work in San Diego, he has identified the expression of T-Box 18 in the mural cells, specifically in the pericyte population, and also at that time, out of coincidence, I would say, when he was checking at different cell types expression, he identified the expression interestingly, so robustly in the aorta, which initiated this project basically. So if I would say, if Nuno can add on to the earlier development of this part, can add
Well,
on, please.
yeah, well, the early development is really related to embryonic development. Basically, it's a gene that is very important as a marker of specific populations of cells during cardiogenesis in embryonic development. And the lab where I did my postdoc was historically interested in this gene under an embryogenesis perspective. The tools were available, and I took it a little bit more to the adult perspective. And when we saw the expression in this cell type that was a little bit unexpected, we decided to explore to see if there were any important roles given the very specific expression pattern. And that's how we got interested in it. It was not the result of any fancy screen or any search approach that we often do in the lab. In this case, was a little bit more an ongoing interest and the availability of tools that we already had at hand.
That's great. Thank you.
Thank you. I think we have another hand raised. Please ask your question. KM, please introduce yourself.
Thank you very much. It was very interesting talk. And I wonder if Debanjan and co-authors have tried to find out the exact pathway, molecular pathway, by which the TBX18 can control and improve aortic development. For example, I have idea because we saw FOS, ATF3, and also GUNB, which are transcription factors by themselves as well, and they are just immediate targets of MAP pathway. So, and there is in literature data that if MAP10 is knocked down, there is a malformation of aorta. Could it be that this pathway is just exactly the pathway which phosphorylates and work with this AP1 complex? Because as I see from FOS increase, it means that FOS and GUNB coming together, they are just switching on survival mechanism. And in this case, there is no much of differentiation. So it is much more proliferation, which brings aorta to become enlarged, but not appropriately for. So that is my idea, and I wonder if Detyan and your co-authors have tried to go further and come up with the pathomechanism.
Yeah, that is a really interesting question, and I would say that the genes that you were mentioning, like ATF3 or FOS and June, like the AP1, also what we see not only from the transcriptomics or translatomics data sets, but also from the human aortic smooth muscle cells perturbation, that obviously due to time constraints and also I did not show in here, but these are also the potential targets like ATF3, EGR1, FOS, June. These are the immediate early genes that TBOX18 directly regulates. Now, coming to the point of proliferation, indeed, in the in vitro model where the cells are more synthetic, we do see increase of proliferation, or which is somehow yeah, can be linked to the survival mechanisms. While in case of the in vivo model, where the lesions are formed in the aneurysmal medial layer of the smooth muscle cells, we have tested at different adult time points, but the smooth muscle cells, we do not see any proliferation at all. And even so, in case of the Marfans or the double knockouts, the cells often lead to death and the disorganization, not like thickening up like we see in other proliferative models, for example, like a TAC model where fibrosis is shown. Here, the smooth muscle cells does not undergo so much proliferation, at least at the in vivo level, what we have seen. One important aspect we can again hint towards is the smooth muscle cells undergo from a contractile phenotype to a more synthetic phenotype, and in that process, I think the T-box 18 is help. Yeah, in this way, presence of smooth muscle cells is presence of T-box 18 is repressing these immediate early genes and thereby prohibiting it going to the synthetic phenotype.
Thank you very much.
Welcome.
Can we have a question from Miran as well?
Thank you. This was very, very, I'm interesting, and I'm amazed by all the methodologies that you have used to really decipher all these stories related around this gene. I'm really impressed by the methodology that you used, especially with the TRAP methodology, how you actually focused on the translatome, as you call them in your presentation. So I'm fairly interested in applying this methodology, at least in theory, and I was wondering how much RNA you started with, because I know when you do complete extractions of RNA, and then if you do the ribosomal depletion, you end up with not very much left. I can imagine what is the amount of mRNA that is attached at ribosome at any given moment, that this is quite a low amount. So I'm just wondering, from the technical perspective, if you can elaborate a little bit more on this, and then if you found any significant difference in compared to bulk transcriptomics, because I saw that you in parallel performed the bulk transcriptomics as well, and this TRAP protocol. So if you can give us some more comments about the differences, what you have observed, because we always see that when transcriptomics is coupled with proteomics, there is not much correlation between these two layers. So I'm just wondering if this novel type of protocol can shed some new light. Thank you so much.
Yeah, thank you. I can elaborate much on this technical aspect first. Yes, indeed, this is a very powerful protocol, and the one that I have shown you is the trap sequencing protocol, which was initially back in 2014, I think, was initially developed for brain studying glial cells and neuron interactions. But we use this transgenic line by crossing it with our gene of interest, and therefore could be used essentially can be used for any particular cell type specific tree. Now, yeah, indeed, the amount of RNA is a critical factor, and luckily, in the aorta, if you see, is a macroscopic big organ, and in our case, we did not restrict only to the thoracic aorta, but the complete aorta, say from the left ventricular junction till the junction to the kidneys. So basically, the whole of aorta, and from that amount of tissue, if you remember that the majority of the cells in the aorta are the layers of the smooth muscle cells, which are multiple layers. So the majority of the cells coming out are mostly the smooth muscle cells. So from the amount of RNA, we were at a good point to start this experiment. It has been quite a while, almost 2 years, if I'm not missing the exact nanograms, but the range what I can suggest is around, I think, 100 to 200 nanograms was enough to do this experiment with. But over the period of time, there has been advancements in the detection of the libraries. So nowadays, the low input libraries are also giving pretty good amount of amplifications. So now even one can even think of having a pilot experiment with even 10 or 15 nanograms also, what the catalog says for library amplification. So this is the first thing that, and the second part was how much it was yeah, overlapping with the translatomics and the transcriptomics. Yeah, that's also a really a good question, and the answer is in two layers, I would say. Firstly, is the contribution of the other cell types. So in case of aorta, as you know, that there will be endothelial cells, there will be the outer adventitial fibroblasts, and also now and then there will be immune cells stuck, which can also contribute to some of the transcriptomics, because in the bulk RNA sequencing these all data will be these all RNA transcripts from these will come in a transcriptomics, while in translatomics there will be not only the data coming out from the smooth muscle cells, but also the actively transcribing mRNAs. So not even the mRNAs present in the smooth muscle cells, the only ones which are actively transcribing, they are contributing. Therefore, the differences are quite strong, I would say, and many of the genes that often have come up in the transcriptomics, only a small subset or a percentage of it we have seen in the translatomics, which is also quite reasonable because yeah, these are the genes which are uh, changing due to the due to the condition. For example, if it's only actively transcribing in a double mutant and not in the yes in the controls. If Nuno would like to add on to it, might also be good.
Yeah, maybe just a very short point. I think I'm not sure I completely agree with it, what you just said, Evangeline. I think so. In terms of the differences, most of what we saw in the transcriptomic level, we also saw at the translatomic level, often amplified, just because we were focusing on a specific cell type. So we were losing the buffering activity of non smooth muscle cells, and we also had because of this for the things that were changing maybe not so strongly accord genotyping groups, we had a higher resolution capacity. So there were things that we saw at the translatome level that we did not manage to see at the transcriptome level. But I think the bunch, I don't remember the numbers exactly, but I think like there's an 80% overlap, depending a little bit on the genotype group, between data sets. But some of the very interesting biology was only seen in the translatome.
Thank you so much.
Thank you both for all the interaction during this Q&A section. I think we have time for a final one, and I can see in the chat a question from Bianca, and she's asking whether you guys can please comment further on the differences in aortic expression in mice and human that you see for a TBX18, because you partly touch upon the sex differences, but if you like to comment further, that'd be great.
Yeah, maybe I can show in the presentation if it's in my in the last images. If you see in the figure number 6, I think A or B, there we have explicitly have shown the contribution of male and female, and there the transcriptomics data that is also shown in our from our collaborator Professor Magnificent's lab. There they have almost equal contribution of male and female in the cohorts. So there also we see similar contribution in the patient cohort. Unfortunately, in the mice, especially in the adult uh, knockout, like in the myosin heavy chain CRE-RT2, we were how can I say that um, we were we could only look at the male mice because the the Cree, that is the myosin heavy chain 11 Cree, was only integrated in the Y chromosome. Therefore, unfortunately, this female mice from this study could not be studied because the Cre was specific to the male chromosomes only. Therefore, for our all the analysis that have been done in mouse work was in males, and therefore to complement it, the cohort that we have used in both the cases had almost an equal fair share of the male and female gender contribution, and we saw similar effects. Yeah.
Thank you for clarifying that. So I would say thank you so much for this brilliant session. It was great to have you guys kicking off this first Journal Club of 2026. If there is, well, there is no time for further questions. So if you guys want to reach out to Nuno and Debajan because you have want to chat more, I'm sure they'll be happy to stay in touch. Just let us know and we can forward their details if you cannot find them. And thank you for joining. We'll see you all for next session. We'll circulate information about the next topic in a few days. Thank you everyone for joining.
Thank you.
Have a good day.
Thank you for having us.
Thank you for being
Thank
here.
you.
Yeah, absolutely. It was great having you guys.
Thank you.
Thank you. Bye, Nuno.
Bye.